Hi guys, Miss Francis here. I would like to start our discussion on osmoregulation. So if you look at the word itself, osmoregulation, it really gives itself away as far as what it is. It's just regulating through osmosis. It's the management of the body's water content and solute content based on controlling the movement of that water and of the solutes between the external and the internal environment. So how are organisms going to actually do osmoregulation? Well, it varies depending on that organism, but the process of excretion involves the removal of metabolic wastes before they accumulate to those harmful levels. Excretion also helps us do osmoregulation. So when any organism does any sort of metabolic function, whether it be cellular respiration, whether it be digestion, some type of metabolic waste is given off. Examples would be carbon dioxide and water, which are both byproducts of aerobic respiration. Water is also a byproduct of dehydration synthesis. Your nitrogenous wastes come from amino acid breakdown, which is known as deamination, and nucleic acid breakdown, right? So deamination is just the breakdown of the amino group. We know that amino acids obviously have an amino group, and your nucleic acids have nitrogenous bases, which will become part of your nitrogenous wastes. So there are three different types of nitrogenous wastes, and we will get into those three different types of nitrogenous wastes. But before that, we must also realize that mineral salts are a byproduct of all of our metabolic processes. And as we know from our study of osmosis and water movement, salts play a role in determining whether an environment is hypertonic, hypotonic, or isotonic, and therefore will influence the way the water is going to move. So here are those three different types of nitrogenous wastes. Ammonia is the first type of nitrogenous waste that I want to discuss. Ammonia is the nitrogenous waste that aquatic organisms are going to produce when they break down their amino acids and their their nucleic acids. However, ammonia has a high toxicity level, but it's also highly soluble in water. So fish are able to flush out that highly toxic ammonia because they are constantly surrounded by water. Urea is the nitrogenous waste that organisms such as humans produce. Its toxicity level is medium, and its solubility in water is also medium. The last type of nitrogenous waste that we'll discuss is uric acid. Uric acid would be found in your flying organisms, such as birds, or your dry land organisms like desert lizards, where the toxicity level is low, and you don't really have that much access to water per se, where you can release this uric acid as a solid. It's that white part of the bird excrement that you might see on your car in the morning if you're parked under a tree or on the boardwalk, because there are lots of birds over by the boardwalk. But in general, the kind of nitrogenous waste, whether you excrete ammonia, urea, or uric acid, all depends on the evolutionary history of that organism's habitat. For example, what was its availability to water? So our proteins and our nucleic acids get broken down into different nitrogenous wastes. The nitrogenous waste that those proteins and nucleic acids get broken down to depend on the external environment that that organism evolved in. So aquatic organisms are going to break down their proteins and nucleic acids into ammonia. Your mammals, which include us, are going to break down your proteins and nucleic acids into urea. And lastly, birds, insects, reptiles are going to break down their proteins and their nucleic acids into uric acid. So here's a nice little review of those metabolic wastes, which you can go through on your own time. Lastly, here's a review of adaptations for excretion. You don't need to know about all of these organisms for the AP exam. You will need to know about the bird. However, for the SAT-2, it would be helpful for you to know about these organisms. So when you have a chance, take a look back and you can study these. All right, that's going to end our discussion for today. Tomorrow we'll continue talking about osmoregulation.